This is a model of the neuromuscular junction. Muscle and nerve are two very unique kinds of cells. The nerve has the ability to generate an action potential and conduct it, and muscle has the ability to conduct the action potential and cause contraction. What you're looking at here is a piece of a single muscle fiber. So a muscle fiber is a muscle cell, but it's a long cylinder and can have many nuclei, up to 100. So we're just looking at a small piece of a single muscle cell or muscle fiber. If we start from the outside, we can see that the muscle cell or muscle fiber is covered by some connective tissue which help bring blood vessels and nerves to each muscle fiber. This is called the endomysium. On this model, the muscle's plasma membrane, called the sarcolemma, is seen here in tan. Each one of these cylinders is a myofibril. And between each of the cylinders, you can see numerous mitochondria. Obviously, muscle is very rich in mitochondria because of the constant need for ATP to facilitate muscle contraction. If we look at the sarcolemma here, we can see that there are some holes in it. These holes are the openings to transverse tubules. The transverse tubules, or T-tubules, are seen here in blue. It's the transverse tubules that is going to conduct the action potential into the sarcoplasm to the sarcoplasmic reticulum. The sarcoplasmic reticulum contains calcium, and in this model you can see it as this tan net-like material. At the junction of a transverse tubule with two end cisternae of a sarcoplasmic reticulum, we call this a triad. So a triad's a transverse tubule and two end cisternae of sarcoplasmic reticulum. If we look over here, we can see one of the nuclei of this part of a muscle fiber. The nuclei are located just under the sarcolemma. That allows the entire sarcoplasm to be filled up with these myofibrils. The myofibrils are made up of repeating units called sarcomeres. The sarcomere is the structural functional unit of skeletal muscle. Each sarcomere extends from a Z line to the next Z line. If you look at the muscle fiber under the microscope, you can see that there are alternating light and dark bands. The Z line goes right down the middle of a light band called the I band. The dark band here, this is called an A band. This part of the A band contains overlapping thick and thin filaments, actin and myosin. The center part, called the H zone, only contains myosin, or the thick filament. This part of the I band contains actin only. When the muscle fiber is given the signal to contract, calcium is released from the sarcoplasmic reticulum, and that binds with components in the muscle fiber, and the sarcomeres get shorter. As each sarcomere gets shorter, each myofibril gets shorter. As the myofibrils get shorter, the muscle fiber gets shorter. And if enough muscle fibers have been stimulated to contract, then the muscle will shorten. Let's take a look at the nerve component. So, nerves control muscle contraction. Without the nerves, the muscles will atrophy. So the nerves are necessary for the correct physiology of the muscle as well as to facilitate muscle contraction. In this model, we can see the end of a nerve. This would be like a synaptic terminal or a synaptic bulb at the end of a myelinated motor nerve axon. What we're seeing right here is a little bit of the nucleus of a cell called the Schwann cell or the neurolemma site. The neurolemma site wraps around the axon, around and around and around, and creates a sheath of myelin. This myelin sheath functions to insulate the axon, and this greatly increases the speed of conduction. The axon itself is the smaller yellow part in the middle surrounded by the myelin. You can see mitochondria in the axon as well. Let's take a look at the details of the junction, the neuromuscular junction. So right here, this is the end of the axon. This would be the axon terminal. And this membrane in here is called the presynaptic membrane. That belongs to the axon. If we look here, we can see that the sarcolemma in this area is greatly folded. This part of the sarcolemma in the neuromuscular junction is called the motor endplate. And on the motor endplate, we have receptors for acetylcholine, the neurotransmitter, which are attached to sodium channels. When a wave of depolarization reaches the axon terminal, these little white vesicles are dumped out into this space called the synaptic cleft. These little white vesicles contain neurotransmitter. In the case of muscle, this is acetylcholine. The neurotransmitter binds to its receptor on the motor end plate, and this opens up the sodium channels. The wave of depolarization caused by the sodium channels enters in to the sarcoplasm, down the T tubules, making its way to the sarcoplasmic reticulum. When that happens, calcium is released and the muscle begins its contraction phase. I think that's it.